back to our discussion. After the acrosomal reaction, the most important step, that means the fifth step, this is about acrosomal reaction. Acrosomal reaction, the fourth step. The fifth step is called sperm oocyte binding. Oocyte binding. Again, just we can rub off certain structures here, like corona radiata and zona pellucida. I told you already. It will create a particular tract, not entire structure will be break down, a particular tract, so that the sperm will, what we call later sperm, will bind with the, the plasma membrane, what we call sperm oocyte binding. Here the sperm will ultimately, bind with the plasma membrane. We will magnify here, this is the plasma membrane of the oocyte, then this is the later sperm. Here part of the sperm membrane will be binding the plasma membrane, because the acrosomal is opened out. The head of the sperm, that means the anterior portion of the head will be detached off. Okay, here the sperm membrane will bind with plasma membrane. Again, it is important here, what we call fertilin and partner protein interaction. Fertilin present in sperm. Fertilin present in sperm, which is nothing but a dimeric glycoprotein. A dimeric glycoprotein. Then corresponding oocyte protein, what we call partner protein. It's like this is fertilin and this is antifertilin. This is another partner molecule. This is fertilin and its partner molecule. Okay. The what we call sperm oocyte binding. What is sperm oocyte binding? The sperm contains a dimeric glycoprotein, what we call fertilin, and this fertilin binds with that of the plasma membrane protein of the oocyte, what we call sperm oocyte binding. This binding is essential because it facilitates a fusion. Then after it binds, it has to fuse. It has to fuse again. Fuse. This is very important. After this, a series of events will be taking place, because till now the guy has been arrested at the metaphase 2, what we call meiosis 2, metaphase 2 of meiosis 2. Then it has to continue the rest of the cycle, say anaphase and telophase, like that, so that the oocyte, the secondary oocyte will lead to creation of a larger ovum or egg and a second polar body. Both are haploid. The third step, this is very important reaction, what we call cortical reaction. The sixth step important. After the sperm oocyte binding, the sixth most important reaction, what we call cortical reaction. These are the two reactions which are discussed at length. Cortical reaction and zona reaction. Cortical reaction. Say we have discussed here that these are the cortical granules, the red color structures. Cortical granules. Here the point is, since the sperm oocyte has been arrested at the metaphase 2, it has to get activated first, so that it can complete the meiosis 2. When the sperm binds with the oocyte, it leads to activation of egg. How and what exactly what makes the egg to get activated? This is very important. What exactly means it will become depolarization. When the sperm fuses with that of the oocyte, it leads to depolarization, what we call depolarization. Depolarization of oocyte. What happens when the oocyte get depolarized is it increases the calcium ions. This is very important. What is the reason, and that means what are the consequences when there is a calcium ion increase? When the oocyte get depolarized because of sperm interaction, it leads to increase in calcium ions, and these calcium ions will lead to cortical reaction. How? It will enable the cortical granules to open out, just like exocytosis, cell vomiting. For example, so these are the cortical granules. Cortical structures will draw here. Okay? These are the cortical granules. Okay, cortical granules, and this is zona pellucida. So next, the surrounding the next layer. The content will become outside. What exactly the content will be present in cortical granules? They are the secretory vesicles contain different enzymes like proteases. We have discussed zona pellucida is made up of ZP3 protein. Here, most important step we have discussed about regarding cortical reaction is block to polyspermy. Block to polyspermy. It is of two ways: fast block to slow block. Why it has to block the polyspermy? We have discussed already. The lead is only one sperm will fuse with the egg. It should not allow the rest of the sperms. So how it will not allow? It should somehow there should be a block. But for the fast block and the slow block. Fast block means it's like random. How it happens? We have discussed when the sperm binds with the oocyte, it becomes depolarized, and this depolarization leads to increase in calcium ions, and this calcium ions leads to exocytosis of the cortical granules, what we call cortical reaction. What is cortical reaction? Exocytosis of the cortical granules, which are nothing but a secretory vesicle, so as to release this content outside the cell. What exactly the contents are? They are the enzymes, say proteases, and these enzymes will break down, actually inactivate the ZP3 protein, which is a compound in zona pellucida. Here, the first important, when the cell oocyte plasma membrane get depolarized, it leads to fast block. Fast block means the oocyte plasma membrane, get depolarized. When it is called fast block. When it gets depolarized, it will not allow other sperm so as to enter the oocyte. Only one sperm, let us already get inside. That means it is in the process of getting inside actually, no? Then it will become depolarized and no more for the sperm interaction with the egg. Then the slow block. How it is slow block? We have discussed cortical reaction, what exactly the cortical reaction? We have discussed already. The cortical granules, which contains a set of enzymes like exocytosis, will release outside like proteases, which will inactivate the ZP3 protein of the zona pellucida so that it becomes hardened. This is very important. Zona pellucida should become hardened, what we call zona reaction. Zona reaction again is of two types, How it is possible? Here the ZP3 become inactivated and the sperm receptors will be destroyed. What exactly? That means cortical reaction leads to zona reaction. This is very important. And how cortical reaction occurs? Because of sperm binding. These are all sequence. We have to discuss a sequence of chain of events. One is essential for two is essential for three, like that. Then here after sperm, then the calcium influx. That means depolarization, calcium influx. Exocytosis, that means cortical reaction. Then zona reaction. What is zona reaction? The ZP3 protein of the zona pellucida will become inactivated so that it becomes hardened. Zona pellucida will become hardened, that leads to slow block to polyspermy. And another reaction and a zona reaction, what we call zona reaction, the first step. The second reaction is called, we have discussed the sperm receptors, species specific ligand, like a receptor ligand, and there's a lock and key mechanism. Here the carbohydrate moiety of zona pellucida will be specific to the sperm membrane protein, and that will be acrosome protein, and these receptors will be destroyed so that there is no more further interaction with sperm. That's a simple logic. No more further interaction with sperm, and no more further sperm fusion with that of the oocyte. So that means cortical reaction leads to zona reaction, and this zona reaction will ultimately leads to the prevention of polyspermy. During this process, the one particular leader sperm will fuse. Here there are two words important, syngamy and amphimixis. Syngamy means when the oocyte, that means when the sperm binds with the plasma membrane of the oocyte, that means fuses with the plasma membrane of the oocyte, is called syngamy. We have discussed already now. This is plasma membrane. The first step here is syngamy. Syngamy, fusion what? The oocyte plasma membrane should fuse with that of the sperm. And this is syngamy. After that, the tail will be detached off. Only that means the head Particularly here the binding will take place the post-acrosomal or the posterior region of the acrosome. That means head will be released inside, the tail will be detached off. This is very important. The head part will get inside. The head part. The head part. The tail will be detached off. Only the head part will be inside. Sperm head, protects cells into the nucleus. The pronucleus, what we call the haploid genome of the sperm is called male pronucleus, whereas the haploid pronucleus of the newly formed ovum or egg is called female pronucleus. And the fusion between the male pronucleus with that of the female pronucleus, the fusion between these two is called amphimixis. Amphimixis. The fusion between these two. Amphimixis. This is very important. And these are very important words. Syngamy, first the sperm have to fuse with that of the oocyte plasma membrane, that leads to depolarization, and the cortical granules, cortical reaction because of influx of calcium, and this calcium, and then it leads to zonal reaction. Anaphase promoting complex APCs are also under the stimulation of these calcium ions. So calcium plays an important role here. Because here the sperm become hyperactive because of Caspar channels, which are selective ionic channels that allow the calcium inside so as to form the flagellum bends, so that the sperm will become hyperactive in the viscous syndrome. This is one step. Second step, when the sperm binds with fused with of the oocyte, it leads to depolarization. That means the calcium will be releasing out, and it leads to cortical reaction. At the same time, anaphase promoting complex that leads to anaphase and telophase, like that. Okay. Then the cortical reaction, the releasing out. Then zonal reaction, the zonal plus will be hardened, and that of the sperm will be broken off. Calcium plays an important role in these regards. So ultimately, the syngamy and the amphimixis. Ultimately, we get the zygote. That means the male pronucleus fused with the female pronucleus. That ultimate form the zygote. And when this zygote forms, were discussed in our previous classes, carpus luteum. This carpus luteum prepares for pregnancy because it will release the progesterone that will strengthen the endometrium of the uterus. If there is a successful fertilization, the endometrium will be prepared for pregnancy, for implantation particularly. Okay, this is about the fertilization. So ultimately the fertilization ends with the zygote formation, and the most important significance in fertilization is regarding the sperm. Only the nuclear part of the sperm will be contributing in the zygote, except the centriole. The centriole we have discussed, proximal centriole and the distal centriole. The proximal centriole present in the depression, a notch-like structure at the posterior end of the nucleus, sperm nucleus, and this will be useful for the formation of spindle fibers for the first zygotic division. So, and apart from these two, the rest of the components, no more contribution from the sperm. That means the rest of the components will be detached off. That no more tail, no more mitochondria, only the head part of the sperm will be sink into the ooplasm and will leads to formation of amphimixis. And again, again for what we call first the decondensation step. Then again the female pronucleus and the male to the fuse, and again they form a nuclear envelope, and they start and the zygote formation, the zygote division will start because of centriole. This is about the significance of fertilization. That means haploid. That means here the diploid is restored. And most important thing is when the sperm we have already discussed, the hyperactive sperm, when it finishes with the oocyte and it penetrates and it get inside the ooplasm, it will release energy. This liberated energy when the sperm get inside the ooplasm is essential for the metabolic reactions of the oocyte. That's because the sperm with greater force it will enter. So then it will facilitate the metabolic reactions of the oocyte. And also it forms, it's like organ formation areas. Organ formation areas, zonation pattern will be done in the ooplasm. And reorganization and redistribution of the organelles is most important, that leads to cleavage. Repeated mitotic divisions in cleavage. These are all what we call post-fertilization events and the significance. of fertilization. Okay, so the fertilization as discussed is chain of event: the approximation of gametes, then the chemotaxis, then the sperm capacitation, then the acrosome reaction, then the sperm oocyte binding, then the cortical reaction, then the zona reaction. The zona reaction again, the sperm hardened become, then the zona plates will become hardened, and the sperm will be lost. During this process, the syngamy and amphibians will take place, ultimately zygote, and only the sperm head, the nucleus will be contributed. The rest of the tail will be detached. Whereas the reorganization of the content will take place with respect to organogenesis. Then cleavage will start. It's about fertilization and in a chain of events. Okay. The most important is fertilizing and anti-fertilizing reaction. They know that, and the acrosome or the zona lysine releasing by the sperm, which break down the sperm, then zona pellucida layer. And most important is hyaluronidase, which break down the hyaluronic acid, and the fettling interaction of sperm with that of the apartheid molecule in the oocyte. This is a specific issue that can maybe asked in the examination point of view. In a cortical reaction that leads to exocytosis because of influx of calcium that will inactivate the ZP3. Okay, and that means cortical reaction is a zonal reaction. It's about a sequence of events that ultimately leads to zygote formation. That means ultimately the diploidy has been restored. Okay, it's about fertilization.